Annotated corpora can be used for several purposes such as information extraction from given data, training of given data, then testing of unknown data. There is another advantage of annotated corpuses, that is classification. What is classification and what are different corpuses available which are used for classification of documents? So initially classification can be defined as it is basically an activity to assign qualitative labels to some given sample data. So what are these qualitative labels? And in some examples these qualitative labels are also attached with quantitative labels too. So example of classification can be, it can be done at different levels such as binary level. Binary level means that we just say yes or no about a data. For example, we say that either this cell is a cancerous cell or non-cancerous cell. Similarly, we have some seed which is germinated or cannot be germinated. Then we have categorical data. Categorical data means that we assign different categories. For example, we say that there are 5 grades A, B, C, D and E. And finally, we have some numerical classification. In numerical classification, we assign numeric values to some data. For example, age of a sample, we can say that it is 2, 3, 4 and so on. So what is the usefulness of these data sets for classification? Finally, again, they can be used for assigning labels and putting annotations to some data. Annotations are our tags which help us to understand some specific data set. Again, after classification we have several steps or levels, such as we have clustering. So clustering is a similar concept to classification, but difference is that we don't know beforehand the label of the given data. So whenever we have some samples, what will happen in clustering, that there would be some blocks or groups of similar data. So let us say if there are 7 samples, then we can have a block of 3 samples in one part and block of 4 samples. So similar samples will join together to make one cluster. Then we have association rule mining. Association rule mining means that what is the relationship between different variables. This association rule mining is again similar to another concept which is basically regression, but the difference is that regression is the relationship between 2 variables, where one relationship is dependent variable and the other variable is independent variable. So what are different data properties for classification? For classification, we should have some data and then we have some domain experts which understand data and they can classify data based on those information. And then this information is indulged into computers and then they mimic the properties of human experts to find out classification patterns in the given data set. Here we have a concept of biomarker identification. So what is biomarker? Biomarkers basically, they are measurable quantities inside data to have some tagging or annotation information. So what is the steps? Number 1, they try to identify key variables, which are the key variables inside a given data stream. Then related to those key variables, they try to find out what are the most related variables to those key variables. And then they present higher prediction values. So prediction values are related to variables, then their conditioned joint parameters or variables, and then they try to predict new values out of those variables. Similarly, this identification between variables, it can be at 2 levels. Number 1 is correlation. Correlation means that they are directly proportional to each other and they have some similarities together. And it can be anti-correlation also. Anti-correlation means that they are vice versa to each other. They are different, and whenever one value increases, others decreases and vice versa. So by acting these together, we can generate new predictions. This can be positive or negative, means it can be correlated or anti-correlated. So now what is BioInfer? So BioInfer stands for Bioinformation Resource. So this bioinformation resource is basically doing 2 things. Number 1, bioinformation extraction of documents, and then this is a resource for these purposes. So bioinformation extraction resource, what does it do? It extracts information from documents and then assigns labels or classifies the new data. So what is this information extraction type of? It works on genes, DNAs, RNAs and different scientific publications which are used to collect this information. So some statistics about this data is there are about 1100 sentences which are taken from biomedical research papers. Then we have two common things or two main things. Number one thing is actual data that is present in the medical papers, such as named entities. Named entities, for example, we have different names of DNA entities, RNA entities or different proteins, and then combination of those named entities with ontologies. So what are those ontologies? Ontologies are upper level of those domains, so that we have complete hierarchy of information inside some medical data.